We're going to continue to talk about genetics, and I realize from your board expressions that a lot of you have had this genetics, but I just want to make sure, because I have a theory, sort of my own theory, that plant breeding is a very simple science. It's very, very easy to comprehend and understand, except that it's a very complex science. And so if you dive right into advanced breeding methods, just the jargon and the terminology alone make it so complex that it's kind of hard to follow what people are really doing. Whereas if you start with the basics and sort of build degrees of complexity, hopefully it'll be a little bit easier to follow. And today, actually, we will cover material that comprises probably 2.5 genetics classes. We'll talk a little bit about mutations, then we'll talk about quantitative inheritance, and later on, the last 2 weeks of the course, you will get a much more mathematical, statistical view of quantitative inheritance when Don Vyans comes in to talk about population improvement and recurrent selection. We'll talk about heritability, ties in with the quantitative inheritance. We'll talk a little bit about population genetics, and again, you can take an entire course on population genetics or genes and populations. Then we'll talk about polyploids. All of these factors make our simple Mendelian inheritance and Morgan's gene zone chromosomes and linkage even more complex. So, you know, it'd be nice if in plant breeding we dealt with only single dominant genes that controlled our important traits. Well, we don't. And so even though all of these genes follow basic laws as defined by Mendel and modified by Morgan, it gets fairly complex. So mutation is a very, very simple concept. It's just a change. And in biology, we're talking about change in the genetic material. But the first thing we must realize is not all of these changes in the genotypes even show up in the phenotypes. So we can have hidden changes until we start looking at the molecular structure. There are two major types: big mutations and little mutations. And within those types, for sort of the big changes or big rearrangements of genes on chromosomes, we have deletions. These are all pretty self-explanatory. Duplications, inversions, translocations, and even transposable elements that sort of jump around the genome. Oops. Just to show you, both the duplications and deletions often arise through uneven crossing over. So again, right back to the basics of meiosis, where homologous chromosomes pair, sister chromatids line up together, and crossing over occurs. If crossing over doesn't occur at just exactly the right sequence within genes, we can get mutations. Translocations I've thrown in because translocations often involve sort of crossing over between non-homologous chromosomes. And so you ask, well, if the chromosomes are not homologous, they're not going to pair, how do you get crossing over? Well, a little bit later we'll talk about polyploids. And some of our crop plants have several different basic genomes so that they have chromosomes in one or more of those basic genomes that are similar, and sometimes these chromosomes get confused and chromosomes that aren't really true homologs match up, and we get and of course transpositions I just throw in here because it's really just movement of small areas. They can move to the same chromosome to their homolog chromosome, to non-homologous chromosomes, and these were first observed in the 40s by Barbara McClintock, who was at Cold Spring Harbor at the time, but who was a graduate student here at Cornell, and published this work in the 40s, and for about 30 years was considered a batty old lady, until finally the molecular technology caught up to Barbara's mind, and people realized that no, in fact, there are jumping genes. And a whole series have been identified and studied. The smaller mutations, two basic types. You can either have the loss or the addition of nucleotides within the gene sequence, or you can have substitutions, so that we can make a mistake and put the wrong purine base or pyrimidine base within the structure. Now, that's sort of your basic introduction and course in mutations. Why do we concern ourselves with wanting to know about mutations in plant breeding? Well, they occur in nature. In general, a locust might mutate 1 out of 1,000,000 times that it replicates. So the sort of the rate's 1 in 1,000,000, plus or minus a few 100,000. They can be induced. Chemicals and various types of radiation, in fact, can cause mutations. At one time, mutations were considered to be the chief source of the origin of new species. And so back in the 30s and 40s and into the 50s, plant breeders decided, wow, all of this variation that occurred and was selected during crop domestication, most of it came from mutation. So if we can find a way to induce site-specific mutations, we're going to have a whole new science, and this science of plant breeding, of making crosses and following progeny, is going to become obsolete. And so back in the 40s and 50s, there was a big wave of people rushing into mutation breeding. Well, after that wave of mutation breeding didn't lead to any real significant successes, people now realize that it's mutation coupled with hybridization that gave rise to the variation that resulted in crop domestication. Now, why would hybridization contribute to that? Why would hybridization increase genetic variation? Well, we get hybrid vigor, but it's ironic because now we consider hybridization to produce hybrid vigor the major factor driving uniformity. So why would hybridization contribute to variation? Speak up. Well, we have this nice mechanism to maintain the same numbers of genes from generation to generation, but the point you're making, if you get different gene combinations together, if you get a hybrid between individuals with vastly different genetic composition, then the hybridization brings one chromosome from each of those parents together to pair during meiosis, and you get crossing over and recombination of genetic material. So one of the big ironies that we face today is hybridization is probably the driving force between most of the creation of additional new genetic variation and variability. But yet, as our farmers and consumers and customers understand hybridization today to produce an F1 hybrid variety results in a very, very uniform variety and very uniform product. So it's a case where, depending on which side of the hybridization issue you're working from, you're either creating a lot of additional genetic variability or you're driving the world to the verge of disaster by creating so much genetic uniformity. Just put these up here. People say, well, did mutation breeding ever work? Yeah, it works. It still works. Our colleagues in Europe are still pounding out new varieties induced by, as you can see, neutrons, gamma rays, or chemicals, or X-rays. And these are successful varieties released and grown: rice, wheat, oats, grapefruit, several grasses, lettuce, beans, and a whole series of flowers have utilized this concept of mutation breeding to develop new varieties. As you probably gather by now, mutation breeding didn't replace plant breeding. Mutation breeding became one set of tools that plant breeders have utilized to create genetic variability. And you also have to realize these are varieties that resulted in sort of useful mutants. And even though we say the rate of mutation in nature is about 1 in a million for any given locus, and we can increase that rate dramatically with chemicals or radiation, still 99% of all mutations either have no apparent effect or they're harmful, even lethal. So it really takes a lot of screening of irradiated or chemically treated progeny to identify mutations that are actually beneficial. So it's a little bit surprising that there are a number of mutants that are very, very beneficial for some of our major crops. The dwarf mutations in sorghum, rice, wheat, barley, very, very beneficial. We couldn't have raised the yields of those crops without those dwarf mutations, because normally those crops grow very, very tall plants, and those plants have a tendency to fall over and lodge before the grains are mature at the top of the plant. And so the harvest losses, or the pre-harvest losses due to lodging for all of those crops, can be tremendous. You put in the dwarfing genes and you get a much higher harvest index. The grain that you harvest is a much larger percentage of the total plant, and so the mutations work. Disease resistance, mildew in barley, yellow rust in wheat, rice blast resistance, and then some quality traits: high lysine in barley, low phytic acid or phytin in barley, and waxy maize, all identified as mutant traits. There is, if you're interested in sort of looking at the wide range of mutants available, there are sites, and I think I have it in the references online, or there's a nice book called Mutants of Maize, and you can go through and look through that. Later on, probably next Friday, unless the weather forecasts suggest that Wednesday might be a better day, we're going to go down to an Emerson garden plot just down below the building here, and hopefully it's still in reasonable shape. There is a sort of a demonstration of the chromosomes of maize, so that they're sort of rows that indicate each chromosome, so you can see some of the different mutations that occur on the various chromosomes. All right, so that's mutation. Why are they important? Well, they do create genetic variability. And why is genetic variability important? That's what keeps plant breeders in business. Plant breeders are in the business to create genetic variability and to identify superior combinations of genes in plant varieties, develop and release those varieties. So mutation's a part of that, albeit a small part. Quantitative inheritance. These are characters that are controlled by more than one or two or three genes. Sometimes controlled by as many as 10 or 15. Usually controlled by somewhere in the range of 3 to 5 to 7. But at any rate, once you get characters controlled by multiple genes, you have trouble following the effect or impact of individual genes. So even though these multiple genes follow the same laws of segregation and independent assortment that Mendel proposed, they follow the same laws of being located on chromosomes and being linked and crossing over that Morgan proposed. Since each gene has a very small effect, you can't just walk through a population and say, okay, this plant has all of these minor genes and this one only has 3 or 4. So we have to have some way to identify gene effects. To sort of show you this in a diagram, I mean, we talked about a single gene ratio or a monohybrid and a dihybrid with 2 genes segregating. Well, this is if you get 3 genes segregating. We talked about that dihybrid ratio being 9:3:3:1. That's phenotypically. If you look genotypically, it would be 1:3:6:3:1. Well, by the time you get to 3 genes, it's 1:6:15:20, 15:6:1. And as you add more genes to that, you get a pattern where if you have a few genes, you get high heritability, and the progeny from a cross and an F2 generation segregate into distinct classes. You know, this is assuming incomplete dominance, right? Because you have parent 1, the hybrid, and parent 2, all in distinct classes. Well, as you increase the number of genes, those classes blur. And you start seeing just sort of a normal distribution of plant height in a population, or of yield in a population of plants. You don't see types that look like one parent, the other parent, or something in between. So as you do that, the heritability of that trait goes down, and we'll talk about how to define heritability. Basically, heritability is a measure of how the progeny of a cross look like the parents. So if the progeny look like one parent or the other is something in between, it's highly heritable. If the progeny don't look like either parent, or they all are somewhere in between, or even somewhere out beyond each parent, it's not very highly heritable. So how do we work with traits like this? Well, we have to use statistics, and we work with various statistical averages or calculations. The mean, which is just the average of all the individuals in the population divided by the number of individuals in the population, the sum of all individuals. The variance, which is the difference between each individual in the population from the mean, and it's squared to get rid of the minus signs. Some individuals will be above the mean and some below. And then it's divided by the number of individuals in the population minus 1. And the standard deviation just takes the square root of that variance to get us back down to a standard number instead of the squares. And the coefficient of variation expresses that standard deviation as a percent of the mean. So basically, if you know those 4 factors, you can calculate the variation, the heritability, the expected gain through cycles of selection. And Don Vyans will talk to you at the end of the course and give you some little mathematical and statistical background for this. And we'll expand the formulas to show you how you can actually determine if you're going to work to improve a trait that's quantitatively inherited. How do you go about it in various types of crops? But for now, since each of these genes has a very little effect, and we're not looking and measuring the impact of each gene, we're looking at averages of many genes, then our phenotype is not that readily discernible, or the genotype is not that readily discernible from the phenotype that we look at. So we know that the phenotype is due to whatever genetic effects we have. It's due to the genes in that individual, but it's also due to the environment acting on those genes, the environmental effects, and the genetic by environmental interactions. So if we had a plant that had genes to be really, really tall, but it was growing in a very, very poor soil with very little fertility, it's not going to get so tall. On the other hand, if we had a plant who genetically didn't really have that much going for it in plant height, but it happens to be growing on a nice, fertile, well-watered plot of soil, it gets taller than it should be genetically. And so we have to sort out then which are genetic effects and which are environmental effects. Then we look at our genetic effects. We find, all right, well, our genetic effects can be divided into three major categories, really two major categories and one catch-all: additive gene action and dominant gene action. And then epistatic or overdominance we sort of lump together as interaction effects. So what are these types of gene action? Additive is just what it sounds like. Each allele adds an incremental value. So that if we're dealing with plant height, a plant with 5 dominant alleles or 5 functional alleles would have a height like this. A plant with 10 would be twice as tall. And it doesn't matter, there is really no dominance relationship. So a plant that has AA, BB, CC and has 6 factors would be this tall. A plant that's got big A, little a, big B, little b, big C, little c only has 3 of those factors, so it's half the height. Whereas in dominance, all you need is one allele that carries, that conditions that trait, and a plant that is big A, little a will be the same height as a plant that's big A, big A. We'll go through this again, and when we start talking about breeding self-pollinated crops where our additive gene effects are very, very important, we'll talk about, so I'll give you some cartoons that sort of show how you sort of add and dice these up. But for now, knowing that additive effects are simply each trait, each allele contributes a certain amount to plant height, to yield, to maturity, to whatever. And dominance, the heterozygous individual looks the same as the homozygous dominant, just like Mendel would have told you. You know, is that a problem? Well, yeah, when we're dealing with improving populations, we'll talk about that later. The fact that you have dominance kind of gets in the way because all individuals, for every locus that's heterozygous in an individual, you can't tell whether it's heterozygous or homozygous dominant. So you think you're selecting on a nice additive genetic variance and your increase is half as much because half of those individuals you selected were heterozygous at a lot of loci. But for now, all we need to know is the variance in a population then, you know, sort of how do we use this in plant breeding? Well, if you've got a population that you know is genetically uniform, a self-homozygous individual that all of the genes within the plants of a certain variety are pretty much identical, and we say, you know, a pure line variety of wheat is probably 98% homozygous, so 98% of the genes are pretty much the same. Well, there's not a lot of genetic variation in that population, So when you grow it in a yield trial, and you do yield trials at different locations, and you see differences in performance of those varieties, you say, wow, those differences are due to environment. F1 hybrids are very heterozygous, but they're genetically identical. So if you take an F1 hybrid and grow it over different environments, you can pretty much say the differences observed in phenotypes over different environments are due to an environment. If you take a population that's segregating genetically, such as an F2 generation, or multi-lines or land races of a crop, and genetically they're very distinct, and you grow those over different environments and you see different expressions of phenotypes, well, then you say, well, part of that expression is due to the genotype, part to the environment, part to the interaction, and part to just errors that you make in actually measuring yield or measuring height or measuring whatever traits you're trying to measure. So that when we look at our phenotypic variation for a population, we have to bring in a genotype variation plus the environment plus an interaction. And of course, when we're looking at that genotypic variation, it involves additive, dominant, and the epistatic and over-dominant interactions as well. So basically, using that knowledge and knowing then that genetically uniform populations sort of give you the measure of the environmental variation, because you can sort of grow out a pure line variety and determine the amount of variability and say that's environmental. So now if I take an F2 and grow that out, then the F2 is going to have that environmental variability, you know, presumably about the same amount as the pure line variety, plus genetic variability. So if I sort of subtract out that environmental variability, I can start to get a measure of how much genetic variability really exists. And so we get back to heritability, the degree to which the values observed in the parents are passed on to the progeny. It's all based on phenotypic values. So what are phenotypic values? What is the phenotype? It's not a trick question. Simple. The appearance. The phenotype is what the plant looks like. Whereas the genotype is what its structure and really structural combination of genes are. So basically, when we look at heritability, it describes the ratio of genetic variance due to the total variance. And we talk about broad sense heritability, which we use as capital H squared. Broad sense heritability is nothing more than saying, okay, if we take the genetic variability divided by the phenotypic variability, we can calculate broad sense heritability. So how do you determine phenotypic variability?
Growing the population in the same condition.
Which population? A uniform parent pure line population, and genetic variability, then you subtract that environmental variability from that uniform population from what you see in a segregating population, and you can calculate narrow sense heritability. Why is that important? Well, again, when we get about a week, not next week, but the week after, we'll start talking about breeding self-pollinated crops. It turns out that this additive genetic variance is sort of the backbone of building improved yield and performance of those self-pollinated or pure line crops. So narrow sense heritability is the additive genetic variability divided by the total phenotypic variability. I'm seeing a change from bored looks to maybe some confused looks. Maybe I'm just misreading it. But we will talk about heritability, and we'll talk about some practical ways to measure narrow sense, broad sense, and we'll talk a lot more about additive and dominant genetic variation. Any questions right now over, yeah?
And when you mention overdominance, what does that mean?
Well, overdominance is a case where you have two alleles that each contribute a different effect. And we'll talk about that particularly. It's very, very important in hybrid breeding for heterosis. And when we get through, after we talk about, we'll spend 2 classes on breeding self-pollinated crops, 1 on breeding cross-pollinated crops, and then we'll get to a point where we'll talk about inbreeding depression and heterosis. And overdominance will become a little more clear to you then. But it's basically a situation where each allele can contribute an effect of that much. If you get a heterozygous locus, heterozygous, then you get a bigger impact than either of those alleles. So it's not dominance, it's overdominance. You know, you say if it's dominance, then the dominant allele works here and the recessive here. And the heterozygous between the two is right up to where the dominant allele is. Well, this is a case where it says, well, we got one allele that could be dominant in some relationships and another that could be dominant. When we pull those two alleles together, we get more performance than either would contribute individually. So it's saying that in order to get the best performance out of your variety, you'd like to get every locus in your entire variety to have 2 different functional alleles and get overdominance. And in fact, a lot of people argue that's what, that's what drives hybrid vigor. It doesn't totally. Epistasis also drives hybrid vigor. So when we get to hybrid vigor, we got to talk about not only relationship between alleles at each locus, but relationship between loci in terms of epistatic relationships. Anything else? Because quantitative inheritance sort of complicates our genetics a lot. I mean, it takes us to the point where if we're a very bright and astute farmer selecting for traits, we can do a pretty good job on simply inherited traits. When it comes to quantitative traits, probably a lot of the time we're going to select plants with bigger ears or more grain and thinking that we've selected a good genetic enhanced variety, when in fact what we've selected are plants that probably were growing in a little better environment. So our rate of gains and selections are going to be slower because we got all this environmental noise sort of slowing our progress. And if you're a farmer and limited to 1 to 3 hectares to select from, you don't have much chance to do replicated selections over different environments to sort of manage that environmental variation. And so it sort of slows your progress. It doesn't stop the progress. These farmers are bright. They still continue with their progress. All right, population genetics. Another complicating factor to using genetics to breed plants. Genes and populations have certain patterns of behavior. They follow Mendel's laws, but when we're dealing with populations, and looking at the origin, maintenance, and the significance of the genetic variation, we find some really interesting facts. Well, first I have to throw out this paradox, because we talk time and time again that genetic uniformity can be bad. Well, again, it's all based on your perspective. If you're an individual plant and you're growing in an environment that maybe has some variability, but pretty much it's the same general environment year after year, genetic uniformity is to your advantage. So for an individual organism, that organism needs the genetic makeup best suited to the environment. And it doesn't need some compromise of diverse genes that could give it flexibility to go to different environments. Whereas a population of plants, the advantage is to have genetic variability. Because if the population has genetic variability, then it's much more responsive to changes in the environment. And so in plant breeding, we constantly fight that paradox. It's nice to have populations we're developing that have that genetic variability and buffering capacity, but it's tough to sell an individual farmer on an individual variety that sort of yields an average of 10% below the best variety that farmer could be growing. Because the individual farmers are saying, I'm not worried about worldwide security and genetic diversity. I want the most yield out of my land year after year after year, and I can do that with a lot less genetic diversity than the whole population of my crop needs to carry. All right? When we talk about genes and populations, we talk about, I hate these, so I don't know how to stop these from doing like that too. Some of them just seem to have evolved that way. So polymorphism means more than one allele at a locus. And generally in population genetics, you have to have, a rare allele must have a frequency of at least 5%, and we just can't measure it. Whereas in molecular genetics, I mean, we can look for levels of polymorphism probably as low as you want to go. And monomorphic means populations, all the individuals in a population have the same allele. So this is looking at each individual locus. If all individuals have the same allele on both chromosomes, it's monomorphic, and so the populations are fixed. Why do we use frequencies? Why do we use alleles? Because the frequencies of alleles in a population is a measure of genetic change. And in fact, what we try to do through population improvement is we try to improve the frequencies of desirable alleles and reduce the frequencies of undesirable alleles. What are the allelic frequencies? It's the percentage of a certain allele that's in a population. And the frequency of allele big A is just the number of A alleles in the population divided by the total number of alleles at that locus in the population. And if we look at this example, the frequency of big A is 15. There are 15 big A's up there. There are 34 total alleles, so its frequency is 0.44 or 44%. If we looked at the little a, there are 19 little a's, still 34 total or 56%. And by design, that frequency must equal 1. So the frequency of the alleles at any locus in a population always equals 1. If we assign P to be the sum of the A alleles and Q the sum of the little a alleles, the frequency of P plus Q is 1. If we had 3 alleles, we had an R allele thrown in, then P plus Q plus R must equal 1. So this is our first sort of complexity by dominance. If we have dominance, we run into problems. Why?
We can't tell for sure what the genotype is. It could be AA or
Exactly. When we looked at those plants a minute ago, we could count the big A's right and the little a's right, but all those big A, little a's would look like big A's to us. So dominance gives us problems. It's not possible to distinguish the homozygous dominance from heterozygous. All right, so Hardy-Weinberg, population geneticist, came up with an interesting theorem. Basically, they say, in a perfect world, in a perfect population, that genotype frequencies and allele frequencies remain constant from generation to generation. So there is no variation, there is no selection. On a worldwide basis, in an ideal world, there is no genetic change. It's all sort of accumulated into a ideal population. All right? Doesn't bode very well for plant breeders, except that Hardy-Weinberg says, yeah, but there are 5 forces that can change these frequencies. And so in the real world, we don't have any ideal populations. Mutation, of course, can change the frequency of alleles. How important is mutation at changing the frequencies relative to other factors? Pretty low. Migration can change frequencies. What do I mean by migration? How do alleles migrate?
People coming into the population or leaving the population?
When people take their selection of wheat or maize and they move it across, they only take a certain number of plants and they move them from where those plants originally were domesticated, and the new population starts a new center of diversity, and so migration changes it, and so plants do move. Plants move all the time, usually carried by people or animals. Small population size. Why would that affect? Genetic drift. What's meant by genetic drift?
Just loss of values of frequency.
Yeah, if you take a small percentage of individuals in a population and continue to intermate those, and you don't have true intermating from all of the individuals, your smaller population tends to collect different frequencies of alleles than were present in that original population, and this is genetic drift. Okay? It's interesting, though, because small population size then sort of goes against this universal population genetics of the perfect population. What do we deal with in plant breeding? Small population sizes. If you can grow out 500 to 2,000 plants to select, you're doing good. And that's pretty small based on non-random mating. If you have individuals in the population that flower and mature 2 weeks before other individuals in the population, then your population will tend to shift to an earlier population and a later population. And because the earlier individuals will not mate with the later and vice versa. So non-random mating. Finally, selection. How would selection change the frequency of alleles within a population? Obvious, I hope. Whether it's natural selection or selection aided by humans, if you identify certain plants within that population that are either desirable and therefore you make sure that you get seed from those plants to carry forward, or undesirable and you cut them out to eliminate them from the population. And so basically the selection is the major factor that sort of drives changes of alleles in population. And selection is what plant breeding is about. Just a couple of other things that sort of confuse people at some times. Any one of those forces can change allele and genotype frequencies. Some are more efficient than others, selection probably being selection by humans being the most efficient. In absence of those factors, there's no change in frequencies. So the population's at equilibrium, but the equilibrium just means no change in allele frequencies. It doesn't mean equal numbers of each allele. Okay, so when a population comes to equilibrium, it could be possible if both P and Q are equal to half, or the population could come to equilibrium at any other value of P and Q. And in fact, normally populations at equilibrium do have different values of P and Q. All right, it's time to quit. We're not going to get to polyploids today. So for our next class, I'll continue here and talk about polyploids, which is another complexity of all of these basic genetic rules that we learned. And then I think that'll work out because I'm scheduled to talk on sort of molecular basis of inheritance, which I find more and more frequently is covered in genetics courses. Any questions on what we covered today? If you need to run, you have permission to go. I'll stay around until Ralph Obendorf comes in. I don't think he's out there today. Yes?
Okay, so you say with the Hardy-Weinberg equilibrium that allele frequencies don't change, but how does the genotype frequencies not change if you don't have equal numbers of P and Q in this relation? How does that not change?
In any given population, it's very rare to have equal numbers of P and Q.
Yes, so when it's not equal, how does the genotype frequencies not change?
How does it not change?
Say it's like you have big A, small a, so that frequency is not going to stay the same, but the amount of big A and small a in the population is going to stay the same.
What it's saying is a population equilibrium, you move to less and less heterozygous loci. Or you move, so that if you only have 2% of your plants that are heterozygous, those will continue to segregate, but 98% of your plants are homozygous, and so the segregation of alleles among that 2% is not going to change the frequency that great on that whole population. I mean, 98% of your plants are going to be big A, little a, and the few percent that are segregating might change it from the value of P from 0.48 to 0.482 or 0.483, but it's not going to vastly change. So what it's saying is that these populations are becoming more and more homozygous, either dominant or recessive. And there are lots of concerns with. In fact, Hardy-Weinberg sort of put that model up as being not the factual model, but the model from which everything diverges.
But then over time, the genotype frequencies do change.
Oh, in at populations, they always change. In fact, that's through selection. There is no population that doesn't undergo selection, natural selection. There are very few populations that don't undergo migration or genetic drift, and mutation. They all undergo mutation. So there are no Hardy-Weinberg populations in the world. I mean, they're just saying, theoretically, if we had an ideal world, you could get a population and it would never change. Well, that doesn't happen. So the important thing is not their proposal that it's an unchanging population, an ideal population. The important thing is what factors actually change those populations. And of those factors, breeders use selection and mutation readily. We use different timing of selection of pollinators frequently. I mean, we choose which plants intermate with others. So we really work on 3 of those factors, and then inadvertently we work on small population size because that's all we measure. So we do everything but genetic drift, and I guess you could argue that a breeder's nursery really shows genetic drift or migration from the land races around it. So the whole concept of plant breeding is how do we modify those 5 factors to improve the frequency of desirable alleles and lower the frequency of undesirable alleles. And we'll be talking about breeding methods starting week after next in terms of how do we actually do this with different types of crops.